In every cell, there will be a control center, known as the nucleus. Inside the nucleus, there are chromosomes. Chromosome is the coiled structure of the double helix of deoxyribonucleic acid, or commonly known as DNA. When the chromosome coil is further extended, genes, which are the regions of DNA that encodes a functional ribonucleic acid, or protein products, can be found. For your information, gene is the unit of heredity. If the chain is further zoomed in, we can see that the DNA is made up of nucleotides. What is nucleotide? Nucleotide is the basic unit of nucleic acid, consists of a phosphate group, sugar, and base. Nucleotides will join together to form a molecule of single-stranded DNA. Normally, DNA exists in double strand. A similar chain of nucleotide will formed and joined to each other by hydrogen bonding of 4 different bases in their specific way. Bases are divided into 2 groups, which are pyrimidine and purine. Purine is a 2-membered ring, consists of adenine and guanine. As you can see, guanine is bonded to the oxygen at carbon number 6 of the 2-membered rings, while adenine is bonded to the amine. Pyrimidine is a 1-membered ring, consists of thymine, which is available in the DNA, uracil, which is present in RNA, and cytosine. All of the components of pyrimidine have different structure. For example, at the carbon number 4, cytosine consists of amine while thymine consists of oxygen. There is also difference between thymine and uracil at each of carbon number 5, which thymine consists CH3 while uracil consists only CH. To form a double helix, the purines must be paired with the pyrimidines. You must remember that in DNA, adenine will always pair with thymine while cytosine will pair with guanine. DNA is made up of 6 smaller molecules. A 5-carbon sugar, called deoxyribose, a phosphate molecule, and 4 different nitrogenous bases, which are adenine, thymine, cytosine, and guanine. As in the picture, the nitrogenous shown is adenine. The structure of DNA is presented by a model called a double helix, because 2 long strands twist around each other, like a twisted ladder. The rails of the ladder are made of alternating sugar and phosphate molecules. The steps of the ladder are made of 2 bases, joined together with either 2 or 3 weak hydrogen bonds. As what had been told before, DNA and RNA is almost similar, whether in its name or structure. So, here I show what make them different. First is the structure. You can see from the picture that DNA is double-stranded, while RNA is single-stranded. Second, is the nitrogenous bases that made up DNA, and RNA is different. For DNA, the bases are adenine, guanine, cytosine, and thymine. The bases for RNA are adenine, guanine, cytosine, and uracil. So, the bases that differentiate between DNA and RNA is thymine or uracil. If thymine is present, it means that the structure is DNA, and if uracil is present, it means that the structure is RNA. The third difference is the sugar molecule in the DNA and RNA. DNA use deoxyribose, while RNA use ribose sugar. The picture shown that the deoxyribose contain less oxygen compared to the ribose. That is why the DNA is called deoxyribonucleic acid. Now we will talk about the DNA structure in how it can be twisted, like in the picture. First let we discuss about structure at twisted double-strand DNA. It contain of sugar phosphate backbone, nitrogenous base, and 2-strand, that make up the double-stranded deoxyribonucleic acid. Actually, all this DNA molecules are linearly the same, in which the bases are linked to each other, through the formation of bonds, between the 5-carbon sugar and phosphate. Thus, we have a sugar-phosphate backbone, from which the bases stick out like ribs. Note that our little DNA, or RNA molecule, like the nucleotides that make it up, has both a 3' hydroxyl, and a 5' phosphate end. The first strand we leave in the 3' to 5' orientation, and the other we flip around, to the 5' to 3' orientation. Bring them close together and hey! They stick.